but will be also recorded later on, will be put on YouTube for public. So are you okay?
Yeah, I think it's all right. I mean,
I don't know if there is any unpublished data, but
yeah, I think most of it is published, but it's much of it is also on the preprint server, so it should be all right.
okay, yeah.
Yeah.
Okay, so Mubarak Sahab, I think we are live now on Facebook, so are we good to go? What do you say?
Yeah. Bismillah.
Okay, so good evening and assalamu alaikum to all JK Scientists online viewers who are joining us today for a very special session, an exciting session we have been waiting for long. So we are joined today by a very special guest, our own Dr. Raiees Andrabi, who has been with JK Scientists for a long period of time. He has been in the mentorship team also for a long period of time, but now that he has been very busy, we really appreciate Dr. Raiees Andrabi, you joining us today very early morning. Today's session is on a very pressing situation. It's on the COVID vaccine development. We have been into a pandemic since last couple of years, 2 years more precisely, and the way forward is only vaccines. And we are joined by the master himself, Dr. Raiees Andrabi, who has been working on the development of pan-coronavirus vaccines. Dr. Raiees Andrabi is Institute Investigator at the Department of Immunology and Microbiology at Scripps Research, California, US. Today, Dr. Raiees will be discussing about his journey, some opportunities he will also discuss for immunology students, and most importantly, he will discuss the efforts that are going on in his laboratory related to the development of broad vaccines. Today, we are lucky to have with us Dr. Syed Mubarak Hussain. Syed Mubarak Hussain is founder director of JK Scientists. He's an assistant professor of neurobiology at University of New Mexico. And we are also joined by Umayya Muzaffar from Malaysia. She's a graduate student at University Putra, Malaysia, and she's working on vaccines and therapeutics. Today's session will be moderated jointly by Syed Mubarak Hussain and Umayya Muzaffar. So I would request Syed Mubarak Hussain to kindly introduce Dr. Raiees Andrabi to our audience. Dr. Syed Mubarak Hussain.
Thank you. Dr. Raiees Andrabi doesn't need much introduction. He is a superstar from Ratnipura, Pulwama, and one of our outstanding scientists at JK Scientists in our JK Scientists community. He is a faculty at Scripps Institute of Immunology, as you said. I just want to highlight a few of his achievements. He has over 50 publications, which is impressive in this young career. It's only 3 years, being a faculty at one of the best institutions. He did his postdoc with Dennis Bouton, who is also one of the famous vaccinologists. And he's in a consortium called ChavID. The goal is to develop vaccines. Specifically, it was focusing on HIV before, but now the present situation led his lab to develop vaccines against COVID-19 vaccine, to develop pan-corona vaccines. He did his PhD at Ames Institute, and also he was a master student at Jamia Hamdard. Is it Jamia? Yeah, Jamia Hamdard. And then his lab is mostly funded with through NIH, R33, and Bill and Melinda Gates Foundation. So they fund his research mostly. He has many awards. I will mention a few. He got Young Investigator Award in 2018 and 15 from ChavID. And he has 5 patents as well. He has published in well-reputed journals such as Immunity, eLife, Nature Communication, Science Translation, Virology, and many other journals in the field. It's my pleasure and my honor to introduce Dr. Raiees Andrabi for today's session. Thank you. Dr. Sob, you can start.
Thank you, Mubarak, and thank you, Khalid and Umayya, for organizing this event, and also for the opportunity to present some of the work that we have done in coronaviruses. And I want to congratulate you all for all the efforts that you're making in terms of mentoring students and guiding them in the right direction, and a lot of efforts that you put on in the JK Scientist organization. So congratulations on that. So I will. Okay, so I'm trying to share my slides.
Yes, please. I have made you a co-host. I should be able to share your screen. Yes.
Okay, can you see my slides now?
Yes, it's here.
All right. So as Dr. Mubarak introduced, and my lab is primarily focused on developing vaccines against very complex pathogens like HIV. That's how I started my career as a PhD student, first at Ames, and then I came here to do my postdoc with Dennis Burton, who is a leading vaccinologist, primarily working on HIV, flu, and HCV. So when the pandemic hit, beginning of 2020, we started using our experience from HIV and leveraging that into developing pan-coronavirus vaccine. So that's the data I will be discussing today. How we can learn from what we have learned for other infectious diseases like HIV or flu, and leverage that information into developing vaccines for coronaviruses. So this is where my lab is physically located in La Jolla, California, right next to Torrey Pines Golf Course. This is Pacific Coast, and this whole area is Scripps. This is Immunology building. This is Scripps Hospital here, and that's where all of the work was done. So in my talk today, I'll be discussing the following topics. One is what are the major immune evasion strategies employed by highly variable viruses, and how rational vaccine strategies can tackle those evasion mechanisms employed by these pathogens, and induce broadly effective or broadly neutralizing antibodies that can protect against those viruses. So that's one. And then I'll discuss about SARS-CoV-2 neutralizing antibodies, the variants of concern, which I'm sure all of you are aware of. And also I'll discuss about emerging SARS-like viruses that are in other hosts, and especially the ones that are that use this human ACE2 receptor and could cross over into humans and cause another pandemic. And what are the major challenges ahead for vaccinologists in general to tackle those complexities. So then I will discuss some of the work that's done in my lab on these two broadly neutralizing antibody sites, S2, which is in the spike S2 subunit of the coronavirus, which is conserved across beta coronaviruses, a family of coronaviruses. And then also RBD, where most of the antibodies are elicited through infection or vaccination. So I'll discuss about broadly neutralizing antibodies targeting these two important regions on this spike, and how those could be used to design vaccines. And then in the end, I'll discuss about lessons for development of broadly effective vaccines, what we've learned from our studies in the field, labs who are working on coronaviruses, and also reflect on what we have learned, as I said, from other complex pathogen surfaces like HIV and flu, and how that information would be important here to design broad coronavirus vaccines for. So there are four major immune evasion strategies employed by these highly variable pathogens. So we have a lot of successful vaccines. For example, we have polio vaccine, measles, mumps vaccines. We have many successful vaccines. And when I say successful, many of the vaccines we have, the efficacy is in somewhere more than 90%, which is remarkably efficient. But why do we fail to elicit similar immune responses against some of the complex pathogens, for example, HIV or flu? Although we have flu vaccines, but they are not as effective as we would like them to be because the efficacy is not that great. And it goes back to the complexity of the surface proteins on these viruses that makes our immune system, it's difficult for our immune system to make responses to those complex surfaces. So the four complexities include sequence variability, because most of these pathogens have error-prone polymerase, especially the single-stranded RNA viruses. And when they replicate, they incorporate a lot of mutations, and as a result, they can generate diversity, so variability, and sometimes they would shift away from immune system. So even if you had an effective immunity against the wild type virus, once it changes, even if you have those antibodies circulating in your body, they wouldn't work because the virus has now changed, has shifted. So that's how the virus kind of evades your antibody-mediated immunity. There's also conformational heterogeneity. So as I said, they have this error-prone polymerase, and sometimes it puts in insertions, long insertions of amino acid stretches that create in their surface proteins again, because that's what mostly our immune system sees before the virus kind of lyses and exposes its inner surfaces. Surface protein is the most important molecule, and I will come to that in a few slides. So this generates conformational heterogeneity, and you can physically, if you have a longer loop in the surface protein, you can physically block conserved protein surfaces and prevent the immune system from accessing those regions and recognizing it. So it becomes, again, ineffective. This is a strategy used by HIV most effectively. And then also glycan shield, that's the third major evasion strategy. So what these viruses do, especially viruses that are in this class 1 fusion protein, spikes expressing viruses, they put a dense sugar coat on their surface protein. And what it does is it, again, prevents accessibility of antibodies. And often you need very creative solutions like long CDRH3s, for example, which are rare in our B cell repertoire, to penetrate the glycan shield so that they could see the underneath protein surface. So it becomes, again, difficult. So because those precursors that could encode for broadly neutralizing antibodies, which would be effective against the virus, those are rare because you need these like really creative solutions to get into the conserved bits that are underneath those sugars. This is a strategy, glycan shield, that very effectively used by HIV, in general by lentiviruses, which are HIV-related viruses. But others is like, for example, Lassa virus also has a very dense glycan shield, and SARS also has a lot of glycans, but not as dense as HIV. It's almost like 50% as HIV. But lentiviruses effectively use glycan shield as an evasion strategy. And then immunodominance, and this is basically the virus tricks your immune system into engaging responses to non-essential epitopes. Like it has B cell immunodominant epitopes that are not important for a virus, but it tricks your immune system, and your immune system blasts an immune response to that, which is not effective against virus. So it works in virus's favor. This is a popular idea for flu, and people think it's operational for HIV surface as well. So how do we deal with these four major immune evasion strategies that these complex pathogens have? And the solution is broadly neutralizing antibodies. So these are antibodies that are generated in our body from B cells, and I will go through a few slides to explain how those are generated. And So these broadly neutralizing antibodies are effective against many, many strains of the virus and many diverse, phylogenetically diverse strains of the virus. And I will show you some examples with S2 antibodies for coronaviruses that can go across even beta coronaviruses, not just SARS or SARS-CoV viruses. So there are many examples of broadly neutralizing antibodies against HIV, HCV, or influenza. And now we have many more examples from broadly neutralizing antibodies isolated from people who are naturally infected with these pathogens or who are vaccinated. So how could we use the molecular information of these broadly neutralizing antibodies to design vaccines? And what we call this as reverse vaccine engineering. Basically, you take information from infected humans that have these responses, and what you could do is you could use this as a kind of a framework to design immunogens, so that then you go back and design vaccines and vaccinate people and see if you could engage those types of responses. The advantage is that you know the end result. If you are able to engage those types of responses, you can generate very effective immune, very robust immunity. So largely, if we summarize what we have learned from designing vaccines for complex pathogens, we could come up with three major principles. And most of this is guided by HIV, because it's the most complex virus that humans have ever seen in terms of vaccinology. We don't have an effective vaccine against HIV as yet. We've been trying hard for many, many years, but we don't have one yet because of the many complexities this virus has. So, but we have learned a lot over the years, and this is the work of many labs in the United States and outside, and throughout the globe. There's been so many groups that have kind of been involved in designing vaccines, and largely you could summarize those vaccine efforts into three major principles, how we can design vaccines. And when I say vaccines, I'm mostly talking about here antibody or B-cell responses, how you could generate vaccines that can induce B-cell responses. There is also T-cell component, but for today's talk, I'll be mostly talking about antibodies and B-cells. So what we've learned from other complex pathogens is that, as I said, because these pathogens use these tricks to evade immune systems, so often there's rare B-cells that could encode for broadly neutralizing antibodies. So what you need to do is you need to engineer immunogens, and we do this through the in vitro directed evolution. So immunogens that can precisely target those receptors because they are rare. So you have to find them, and if you get a chance to see those B cells, your immunogen needs to have a property that it effectively engages those B cells to generate an immune response. So what we do generally, we increase affinity or avidity to in our immunogen so that it binds to those B cell receptors effectively. So that you give yourself the best chance once the immunogen sees those B cells, which are extremely rare, but it generates a B cell response, because affinity and avidity is kind of related to a competitive fitness when you go into germinal centers, and then I'll explain that in next couple of slides. To effectively generate an immune response, you need to engage those B cells effectively. So once you've generated those B cell responses, memory B cell responses, then in order to make it more robust, you need to accumulate mutations, more mutations. So that's why we do multiple vaccinations. We prime immune system to activate the B cell responses, and then boost those responses. And for boosting, it's extremely important that we focus on the B cells that are important. Otherwise, we could be distracted by non-important B cells, which are off target. So that's where the second strategy comes into act, immunofocusing B cell response and reduce off target. So we have to we generally use a lot of resurface engineering here again, or we use genetically related viruses that share minimal number of important critical epitopes. And if we use those in sequential immunization, then you could effectively reduce off-target responses and focus on more important on-target responses. And then also the third principle is designs that mimic native-like protein configurations. So as I showed those cartoons on my previous slide, the surface protein is, it's mostly a spike structure, which is in a pre-fusion conformation when you look at the surface of a virion. But once it attaches to host cells, it triggers conformational changes, and that leads to collapsing of the surface protein because the virus has to now release and inject its genetic material into the host cell. So it changes its conformation, and it's called post-fusion conformation. But ideally, in a vaccine, you would want it to be in a pre-fusion conformation because that's the more native configuration you will see on the surface of a virion of an infectious particle. So, for example, I think many of you might be aware of this 2p mutation in current SARS-CoV-2 vaccines, whether it's from Moderna or Pfizer or other companies. So you put 2 mutations in the S2 subunit of SARS-CoV-2 spike, and those mutations trap the trimer in a pre-fusion configuration so that it's more native-like. And we have many other similar ideas in HIV field. We introduce similar mutations. The whole idea is that if you can trap it in a pre-fusion configuration, it will expose neutralizing rather than non-neutralizing epitopes that are exposed on non-native configurations. So just keep those 3 principles in mind, and I will be highlighting these as I go through the data on SARS-CoV-2 vaccine strategies. I don't know why this thing is popping up repeatedly. So let me move. Okay, so this is just to summarize the types of vaccines we use. This is kind of a very basic general idea. What is the nature of vaccines? How do vaccines look like? The simplest of the vaccines are polysaccharide vaccines, and they're sometimes conjugated with some carrier proteins, toxins. So these are the simplest of the vaccines. And conventionally, if we look at classical vaccines, we have had whole pathogen vaccines, and these are mostly inactivated, chemically inactivated or heat-killed vaccines. For example, Covaxin that's used in India by Bharat Biotech, it's like an inactivated vaccine. You basically treat the virus with a chemical, and this is a classical way of doing vaccines. And then you could also use some strategies mixing some parts of human virus versus some parts of the virus that infects a closely related species that the viruses resemble, and you can use some features that make it less infectious in humans, but you could still use some properties that are important for generating an immune response. So those are in this reassorted live vaccines you can generate. And more sophisticated vaccines, and I would say like in last 30 to 40 years, we've been kind of relying on this part here, and this is more recent here. So subunit vaccine is, the idea is rather than using the whole pathogen, you just take a part of the pathogen. In this case, as I said, subunit, the surface protein on these pathogens is kind of what our immune system sees mostly. And if our immune system is effectively able to block it, the virus wouldn't be incapacitated and it won't be able to enter your cells. And that's kind of the idea here. You take the part of the virus, which is the surface protein, you could engineer it in different ways and then express as a recombinant protein. This is the area we work mostly on. And some of the examples here are pertussis vaccines, hepatitis B vaccines, and influenza vaccine. Those are based on subunit vaccines. So you take part of the virus surface protein, engineer it in a way so that it exposes the right kind of epitopes, or sometimes you don't need to engineer because some proteins are well behaved already. And then you could vaccinate and generate an effective immune response. And more recently, this nucleic acid technology has come into act, like mRNA vaccines or even DNA vaccines, and they are rapid and they're fast, and that's kind of one of the major advances why we have been able to so rapidly respond to SARS because of this technology, because you don't have to develop any cell lines, because generally you have to develop cell lines that can secrete these vaccines. You could just synthesize mRNA, inject it in, and your body just makes the vaccine, and then it evokes an immune response. So how do these vaccines work? Just as a curiosity question, once you inject vaccines in, whether it's a protein or mRNA or other platform-based vaccine, how does it work? So there are different routes of immunization you could use, and I have shown here, for example, an intramuscular injection that you get with a vaccine, which is shown in this this cartoon as this orange box here. So once you inject vaccine, so let's say intramuscular, it's taken up by lymphatic vessels, and it's trafficked to your neighboring lymph nodes, for example, or any secondary lymphoid organ. And the example here is lymph node. So these are the structures where you start generating these responses to vaccines, or even infection. If you get infected, your lymph node has loads of virus or bacteria that generate these types of responses. So the main players that generate this reaction are B cells that are able to make these antibodies, T cells that provide some help, dendritic cells that present the antigen on the surface and to generate the B cell response. And the soul of the vaccine, this is how the vaccines work. And this is kind of an important slide to understand how the B cell response is mainly. As I said, T cell response also contribute to protection, but it's more in infection. Most of the correlates of protection are antibodies, and antibodies are generated through B cell responses. And this is kind of the soul of where these responses are generated. So please pay attention here. So as I said, once you inject the vaccine, it gets trafficked to the lymph nodes or other secondary lymphoid organs, and where it's taken up by antigen presenting cells and dendritic cells, shown as an example here. So this is an antigen. The dendritic cell presents this antigen to B cells. So these are the receptors on B cells, which are antibodies. They are in IgM configuration initially, and then once the B cells start making antibodies, they can secrete them as IgGs or other Ig isotypes. So once this interaction, so this is the first signal. Once this interaction happens, B cell gets activated. And then B cell moves to the T cell zone. So this is still in the secondary lymphoid organ, where it interacts with T cells, and it gets second and third signal by interacting through CD40 ligand, and they start secreting cytokines. And so these three interactions define the fate of B cells and define what type of B cell or antibody response will be generated. Initially, the B cells could move to extrafollicular spaces, and they can start making antibodies through these antibody secreting cells called plasmablasts. This happens within a week after infection or vaccination. So when I explain this is more general for both vaccination as well as infection. So these antibodies can be IgM, they can be also IgG, less of IgG, because the class switching mainly happens in these microanatomical structures called germinal centers. So if it goes out, this happens within first 7 days. The B cells can also move to these structures called germinal centers, where they shuffle between dark and light zone for selection for higher affinity maturation. So wherein they compete with other B cells in order to recognize antigen present on follicular dendritic cells and T cell help at the same time. So they shuffle between dark and light zones. dark zone, they proliferate here, and in the light zone, they get selected for higher affinity and higher T cell health. So, and depending on this, so the first pathway was they can directly move into your extrafollicular periphery, but then these two pathways generate long-term responses, and those are, one is they can generate plasma cells, and plasma cells are basically cells that express a lot of mRNA encoding for antibodies, and they get deposited in your bone marrow. So all the serum antibody titers you have in your blood, they come from these plasma cells. They're generally, you don't actually need a lot of plasma cells because they can generate tons of antibodies. They express at least 100 or 500 times more mRNA than a regular B cell, because they're kind of their job is to make a lot of mRNA and secrete a lot of antibody. So these are the B cells that are deposited in your bone marrow, sometimes for lifelong. For some of the pathogens or some of the vaccines, you could get these B cells that sit in your bone marrow forever. And these are responsible for protecting you from reinfections, because now your body can make these antibodies, they're in your circulation. If you contract that virus again, because your immune system has those antibodies that can fight those viruses, that's what gives you protection. The only issue, as I mentioned earlier, some of the evasion strategies that these viruses have developed is they generate variability. Now, if your antibody recognizes the wild type virus, even if you have immunity for that, but if the virus has changed considerably, then you won't be able to recognize it. So even if you have the immunity against the parent virus, you wouldn't be able to effectively neutralize the variant. And then the other type of response is called memory B cells. These are quiescent B cells, and this is more like an investment. So you basically invest into B cells. They just sit around. They don't do anything. They don't replicate. They don't proliferate. They are quiescent cells. But what they do is they recognize the antigen. If you get the same infection the next time, these memory B cells, because as the name suggests, they recognize and remember that pathogen. So instead of going through this process, generating a de novo B cell response, these B cells can spring into action immediately and start secreting antibodies and turn into plasma cells. So it's kind of an investment that you deposit these in your body, and when you have a reinfection or a re-challenge, these can effectively generate responses. And the good thing is that they generate these responses quickly and more effectively, because then they can accumulate more mutations quickly and be more effective. So that's kind of where this, how the vaccines work as for the antibody-mediated immunity is concerned. So what are neutralizing antibodies? And I've been talking about neutralizing antibodies, broadly neutralizing antibodies, and I just wanted to give an example in context of SARS. So this is a cartoon structure of SARS-CoV-2 with spikes decorated on its surface. And as I said, these spikes are important for mediating interaction with host cells and injecting its genetic material into host cells. So for example, this S protein or spike, it has a receptor binding domain, which is around here at the tip of the threefold axis of spike. It interacts with ACE2, which is a receptor on human cells, through this receptor binding domain. And then if it interacts in the absence of antibody, the viral membrane fuses with host membrane, it injects its genetic material, makes more copy of its genetic material, replicates, proliferates, and makes more copies of the virus. So that's why you get infection. But now if you have a neutralizing antibody that blocks this interaction with the ACE2 receptor, you could effectively neutralize this virus and incapacitate it. There are other mechanisms of how you can incapacitate viruses, but I would just focus on neutralizing antibody aspect. And also antibodies can work through the FC portion, and neutralization is mostly mediated through the Fab part, the variable part of the antibody arm. but there are FC mediated, there are receptors on our cells that can mediate FC mediated functions like effector functions like ADCC, antibody dependent cell cytotoxicity, ADCP, CVI, many other functions. So those are mainly mediated through this end of the antibody. So as I said, this spike, so this is the architecture of this spike. It has two subunits, the S1 up here, which is membrane distal, and a membrane proximal S2 subunit. It's a heterodimeric trimer. And as you can see, the breakdown of different domains of this protein, this is the side view and top view for this spike structure. This is SARS-CoV-2 spike. And it's broken down into different regions, and mainly RBD and S2. Those are the regions where our neutralizing antibodies target. And this is the sole target of neutralizing antibody on the virus. And interestingly, and obviously this is kind of expected if we go into other vaccines that we have, the successful vaccines we have for humans, broadly neutralizing antibodies, once you vaccinate people, they correlate with efficacy, protection. And this is true for both vaccination as well as infection. And there are many studies that have kind of demonstrated this. So this is called correlates of protection, and antibodies correlate with protection against SARS-CoV-2. So beginning 2020, when the SARS pandemic hit, we started this collaboration. This was kind of a collaboration with Scripps and IAVI, International AIDS Vaccine Initiative, using this pipeline that we have developed over the years of rapidly generating antibodies. And so basically what we do is we recruited SARS-CoV-2 or COVID-19 convalescent donors and isolated neutralizing antibodies from those in using this pipeline. So this is a very rapid pipeline. We could do this in 10 days, starting from collecting the samples and generating these antibodies, testing in different in vitro assays. And we showed protection in this hamster model that if you infuse these antibodies, you could protect. And it's just kind of a nice proof of principle that if you generate these types of antibodies, they should be effective in curbing the infection. So this was our first collaboration on generating these neutralizing antibodies, and we were not the only ones to do this. There are many labs in US and outside also that have generated similar types of antibodies, and most of the first wave of antibodies that was isolated was against this receptor binding domain, RBD, as I mentioned earlier. And you could broadly classify those neutralizing antibodies into 4 categories or 4 classes: class 1, class 2, class 3, and class 4. And this is based on where they bind to the RBD. And this is based on work from many, many labs. As I said, our antibodies also fall in these categories. But so this is this cartoon in gray shows the RBD part of that spike that I showed you on the previous slide. And based on where these antibodies bind, as I said, you could classify them into 4 classes. Class 1 antibodies bind to this receptor binding site, RBS, where the ACE2 binds. So this is the region where the spike binds to the receptor on the surface of cells. And so class 2 also binds to this overlapping ACE2 site. And then class 3 slightly on the backside of the RBD, and class 4 to this more cryptic. If you look at the trimeric structure, this is more kind of an hidden region. But these sites, the ones that are recognized by class 1 and class 2, are highly variable. And if you see in the SARS-CoV-2 variants of concern, these regions change a lot here. And for other sarbecoviruses, like SARS-like viruses that infect other species of animals, they also have, there's more diversity in this region than this region. So in terms of a vaccine, if you want to target, you would want to be around this region, because those antibodies are more effective, they will be more broader, and they will be long lasting. For example, the current variants of concern have more mutations here than this region. There's hardly any mutations here, because it's always like the conservation kind of falls back to how much can you tolerate as a pathogen to change in a certain region. So these regions haven't changed much in SARS-CoV-2 variants of concern, and also they're conserved across sarbecovirus, other SARS-like viruses. So, so if you want to induce antibodies, this is where you want to induce antibodies to, because those are kind of pretty conserved regions. But unfortunately, if we look at B-cell repertoires from SARS-CoV-2 infected or vaccinated, SARS-CoV-2 infected or vaccinated humans, most of the responses are encoded by VH353. So these are the B cells that encode for neutralizing antibodies, and these are the germline genes that encode for those B cells. And if you summarize all of these studies, 3-53, 1-2, and also 363 to some degree, but most of those antibodies that are elicited by infection or vaccination, they target this part of the region, which is not a great news. Obviously, there is scope. You could definitely mature those antibodies and make them broader. But to begin with, it's the most immunodominant region. So the first wave of your antibody responses, whether be it infection or vaccination, is to this region, and it's pretty variable. And so, so in other words, I can tell you that the most neutralizing antibody responses that you generate in infection and vaccination are to this region. So obviously that's kind of the success of the SARS-CoV-2 vaccines. They were highly efficacious to begin with, more than 90%, but as the variants started emerging, especially Omicron, now those are not that efficacious. It goes back to both the types of antibodies you make, and also are those those antibodies maintained at a protective level. So that those are kind of the concerns. But in terms of vaccine design, you would definitely want to minimize any antibodies that target more variable regions and focus more on the conserved regions. So this is kind of summarized in this elegant study that was published from Ian Wilson's lab here at Scripps, where they took these 4 classes of antibodies and they call them RBS A, B, C, and D, and some of them are classified differently. So based on these 2 different publications. So if you mutate this residue 417, which is part of the RBD, if you mutate that residue to from K to N, you lose activity to those class 1 antibodies that I was mentioning, the most immunodominant antibody response you elicit in infection or vaccination. Completely lose neutralizing, just single mutation. And then if you mutate this residue, it's again in the RBD, E484 to K, you lose class 2 and sometimes other classes as well. And then if you look at those 2 positions, those recurrently happen in variants of concern here, alpha, beta, gamma, and delta. So 417 is mutated in beta and gamma, and then 484 in 3 out of the 4. And if you look at Omicron, I don't have that here. Both of these positions, 417 and 484, are mutated. So in a sense, you would make most of the responses. I'm not saying like all of the responses, because there's always room for affinity maturation so that you could counter escape, but most of the responses would escape Omicron, the class 1 and class 2 types. So that's one problem. How do we deal with variants of concern? Because they're emerging fast, and they're emerging. It's happening in real time, and there's more chances that we'll see more variants in future. So that's one issue. The second problem is that SARS-CoV-2 is not the only coronavirus that has crossed into humans. And we know there are additional 6 viruses, including SARS-CoV-1 and around 2002, 2003, MERS-CoV around 2012. And then there are 4 other coronaviruses that are endemic. They cause common cold like HKU1, OC43, like SARS-CoV-1, SARS-2, and MERS. They belong to beta coronavirus. And then we have also alpha coronaviruses, HCoV, 229E, and NL63. These 4 viruses are endemic, and they cause common colds. They contribute around 15 to 20% of the common cold caused in humans. So the issue is that there are many of these sarbecoviruses or SARS-like viruses in multiple hosts. As you can see, different lineages, SARS-like viruses that use ACE2, and it's easy for these viruses to cross over into humans because they can easily adapt, and multiple hosts. So we have a twofold problem. One, how do we deal with SARS-CoV-2 variants of concern, which is kind of pressing right now? But also, how do we deal with any potential future pandemic viruses as part of pandemic preparedness? So that brings me to the work that we have done. I hope I'm not running over time. Okay, I'll try to go through this probably quickly. So the question is, can we now target conserved epitopes to develop pan-coronavirus vaccine strategies? And it largely relies on 2 steps. One is, can we identify epitopes on the spike? And how can we develop strategies vaccination strategies that can induce such responses by vaccination? So I have 2 topics here. One, stem helix, broadly neutralizing antibodies, and broadly neutralizing antibodies that we have isolated from infected vaccinated human donors that target this RBD region, conserved regions of RBD. So stem helix BNAbs. So if we look at the spike structure, and if we make a matrix of how conserved the regions are, and if we break this down into this identity matrix based on, so red means more conservation. In the S2 domain, which is down here, you see more conservation as compared to S1. And it's mostly pronounced across the beta coronaviruses, like these 5 here. You can see more red, orange. So it means more conservation down here. So we hypothesize that if we look into SARS-CoV-2 infected donors or COVID convalescent donors, can we find B cells that target those S2 regions? So we sorted B cells from 8 different donors based on some earlier analysis we had done on these donors. And if we look at the B cells, memory B cells in these donors, and this is a plot showing percentage of IgG B cells that are specific to SARS-2 spike, HKU, which is a beta coronavirus endemic spike, and also alpha coronavirus NL63. So you see all of them make, as you would expect because of infection, they make a pretty reasonable amount of B cell response to SARS-2 spike. Many have cross-reactivity to HKU, but less with NL63. This is relatively more distant from these 2. Both of these belong to beta coronaviruses, and this one is alpha coronavirus. But then we were kind of more interested in this population. Do we have individuals that have B cells that cross-react between SARS-2 and HKU, and potentially to that base region that's conserved across beta coronavirus? And in fact, yeah, there are some individuals. It's not a huge fraction of B cells, but still pretty reasonable. And we wanted to isolate monoclonal antibodies from these donors. So we selected 4 donors, isolated 20 monoclonal antibodies from these 4 donors, CC9, 10, 36, and 40. And all of those monoclonal antibodies bind to SARS-CoV-2. This is based on a binding assay to the spike. Almost 50% bind to SARS-CoV-1, 1 binds to MERS, and 5 antibodies bind to these endemic HCoVs, and this was kind of promising. These are, again, beta coronaviruses, but none of those showed any binding to the alpha coronavirus, which is consistent with the previous slide that I showed that plot of. So one of the antibodies that caught our attention here was this antibody called CC40.8 down here from this donor that not only showed binding to SARS-CoV-2 and 1, but also to HKU1 and OC43. And then when we tested this antibody against other sarbecoviruses like SHC014 and pangolin-derived virus or WIV1, they effectively neutralized these viruses. So there's the neutralization profiles are comparable. So this is, if you titrate the antibody, and this is the inhibition that we see in our in vitro assays. So they're pretty comparable. They're not as potent as you see those RBD neutralizing antibodies that I showed you on my previous slides, but they can neutralize, and the neutralization is broad, unlike the RBD antibodies. And then if we look at the variants of concern now, they're equally effective against variants, because this region, as I'll show you, doesn't change in the variants of concern. It's pretty conserved there. And we have tested this antibody and more antibodies to this region with Omicron, and they are equally effective against Omicron as well, because this region, as I said, it doesn't change in the current variants of concern, SARS-CoV-2 variants of concern. So then in collaboration with Andrew Ward's lab here, who is an expert in cryo-electron microscopy, we imaged this antibody Fab in complex with an S protein from HKU1, and it shows that it binds to the base of the trimer. As you can see, the flexible, this antibody binds to this flexible region as the Fab samples in different projections. So this is a very flexible region. So we couldn't do a high resolution cryo-EM structure because of the flexibility of this region. But then we did a crystal structure. So we screened and identified which part of the S2 does this antibody bind. And then in collaboration with, in Wilson's lab here, we did a crystal structure. This was done by Mung in Ian's lab, who is a postdoc in Ian's lab. So what this shows you here is this antibody CC40.8 Fab in complex with SARS-2 stem peptide, which is shown in green here. So it adopts a helical conformation in the stem peptide, which is at the base of the trimer. It's part of the fusion machinery of the trimer. So the antibody shown here in heavy and light chains, both of those make extensive interactions with this peptide mainly through hydrophobic interactions, but there are other shape complementarity, hydrogen bonds, and other interactions are also important. But majority of the interactions are through hydrophobic residues, and also this region is heavily enriched in hydrophobic residues, this stem helix region. So then if we look at this region where this is located, as I said, it's at the base of the trimer. It's right. So this would be viral membrane here. And then this is the first, this would be a region proximal to the viral membrane. And that's where the antibody binds. And if we look at that region now, so this is a 25-mer peptide. And if we look at those 25 residues across sarbecoviruses like SARS-like viruses or beta coronaviruses, it's pretty conserved. It's especially the residues that this antibody contacts that are highlighted here in blue. Those are pretty conserved across beta coronaviruses, and that's why they bind to not only to SARS but also to HKU1 or OC43, or even we can go even as distant as mouse hepatitis virus, which is also a beta coronavirus infecting other animal species, but these residues that are important for contacting this epitope are conserved in that virus as well. So that's a promising target. So with this we identified a new target on coronavirus, and this adds more options in terms of vaccine design, but also in terms of if you want to use cocktail antibody interventions, antibody-based intervention strategies. So then the question is, are these antibodies protective? I mean, they neutralize in vitro assays. So for this, we do in vivo protection or challenge experiments where so this is the design of the experiment. We infuse the antibody intraperitoneally 12 hours before we infuse the virus. We inject the virus. We challenge these with SARS-CoV-2. These are human ACE2 expressing transgenic mice. And so we infect, we infuse the antibody followed by challenge. And then we monitor these animals for 5 days. We monitor for viral loads, weight changes, and other clinical aspects that would indicate any kind of disease progression. And then at day 5, we sacrifice these mice and collect lung tissues to look at viral loads and see if the antibody made any difference as compared to the control. So in this experiment, we wanted to titrate this antibody and see if it works at different concentrations. We started with 300 microgram per animal down to 10 microgram per animal. And then we used an RBD CC12.1 antibody, that's a neutralizing antibody, and a Zika unrelated antibody as control. So this is a positive control and this is a negative control. And as you can see from weight changes, you see a considerable loss of weight in the Zika antibody treated animals upon SARS challenge compared to these antibodies. And remarkably, the antibody was even effective at 10 micrograms, which we didn't expect. We were kind of the experiment was designed to see the difference, but we were really surprised that it could protect even at a really low level, despite having a low IC50 neutralization titer. And this is a phenomena we have seen with some HIV antibodies that bind to the base of the trimer, like membrane proximal external region. So then if we take those tissues and look at viral loads, or if we try to grow those viruses and look at plaques, we can also look at replicating virus in these tissues. And in both these assessments, we can see a substantial reduction of viremia in antibody CC40.8 antibody treated animals as compared to the control animals. And this was consistent. So this is CC40.8. This was our first antibody that we isolated for this region. And then we've kind of followed up. And the question is, since this region is conserved across beta coronaviruses, can it also protect against other beta coronavirus challenges? Because in this case, we just tested SARS. So we have isolated, so CC40.8 antibody effectively neutralizes sarbecoviruses and binds to beta coronaviruses, but it doesn't neutralize MERS, for example, because there's one residue change in MERS that this antibody is not good at recognizing. If you mutate that residue on MERS, it does bind and neutralize that, but not the wild type MERS. But then we have isolated more S2 antibodies in last year or so. And I'm showing you 2 more examples of antibodies, CC68.9 and CC99.3, 2 new STEM antibodies that we have isolated recently. And in collaboration with Ralph Barrick's lab at University of Chapel Hill, we have challenged these, we have tested these antibodies in a challenge model with a mouse-adapted virus encoding for SARS-CoV-2 and MERS. And we can see a pretty decent protection in those animals against those virus challenges. So it shows the proof of principle here is that if you could generate these antibodies that protect you, if you can induce these antibodies with vaccination, those should give you broad protection against beta coronaviruses. We have also tested against SARS-1, so that basically tells you that these types of antibodies can effectively neutralize or protect against all 3 severe pathogenic coronavirus that have crossed into humans in the last 20 years, which is remarkable. So this is a very conserved epitope, and these studies show that if you can induce these antibodies through vaccination, they should give you long-lasting protection in terms of even if the virus changes, this region seems to be pretty conserved. We have taken it one step further, like how do we translate this information into vaccine design? And we've generated this nanoparticle where you present repetitive copies of this epitope, this stem epitope. This is ferritin, which is a nanoparticle that has 24 subunits. And this is, again, EM images from Andrew Ward's lab here at Scripps. And this shows a nice profile on size exclusion chromatography, nice peak here. And then in terms of antigenicity, it does bind effectively to this stem antibody. We've tested it with more antibodies now, but this is kind of showing that the epitope is presented in a favorable manner here. So, and then the conclusions from this part is that we can, stem antibodies, they're not frequently elicited in natural infection, but if we can generate these responses by vaccination, that would be terrific. And we could design vaccines based on this stem epitope. One of the things that I kind of highlighted already was that we were surprised that these antibodies can, even at this low IC50 neutralizing, they can effectively protect against virus challenge. And we're pursuing these strategies. There are some issues that we are kind of trying to tackle, like as I said, these are the potency is weak, the accessibility of this epitope could be a challenge, and there might be some B-cell related tolerance issues once you induce these antibodies, and we're looking into those. So those are some of the challenges here. So for the second part, and I'll try to wrap it up quickly now. And so I'm going to discuss about RBD, broadly neutralizing antibodies from infected vaccinated donors. So infected donors make neutralizing antibodies. If you are vaccinated, you make those as well, probably slightly better than that. But if you are infected and then vaccinated, you make much more effective neutralizing antibody responses that can cross-react with other sarbecoviruses and also against the variants of concern. And we've tested this with Omicron as well. So it's still effective against Omicron. If you are infected and vaccinated, you make much more robust response, neutralizing antibody responses compared to vaccinated or infected only. So we saw this as an opportunity, and we thought, okay, can we isolate broadly neutralizing antibodies from these donors? And we chose two donors, CC25 and CC25 and CC84. And in this study, we isolated 93 antibodies, 74 were neutralizing SARS, 65 SARS-1. And if we test this panel of sarbecoviruses that use ACE2 receptor, we isolated 40 broadly neutralizing antibodies in this study. And some of the examples are shown here. They're encoded by a diverse set of germline genes, and they're highly potent against these sarbecoviruses that we tested. These are IC50 neutralizations. And then one of the questions is, are these antibodies effective against Omicron? And this is kind of highlighting, some of the antibodies that we've tested here against wild type, Wuhan strain, alpha, beta, delta, and gamma, and also Omicron. Some of those antibodies could be equally effective against the Omicron variant because they target a region that's conserved across sarbecoviruses. They can effectively bind to RBDs from multiple clades of sarbecoviruses. These are the ones that use ACE2. These are non-ACE2. We don't have neutralization assays for these, but these antibodies can still bind effectively. This is based on SPR binding curves. These are dissociation constants shown coded by colors here. And then again, as I said, we map these. I'm not showing the structural data, but we have done a lot of structural studies, and these antibodies target this more conserved part of RBD epitope and not this more variable part. And again, in some proof of principle studies, we have shown that these antibodies can effectively protect against SARS-CoV-2, SARS-CoV-1, and this SARS-like virus that infects, that's non-human origin, SHC014. Effectively, they can protect against weight loss, reduce lung viral titers, and other clinical parameters. So these antibodies would be, again, very promising, as you can see that even with so many mutations on Omicron, these antibodies are still effective against Omicron because they target the more conserved bits on the spike. So what do we learn from these? And one of the important things that's kind of noted by us and others is that something unique with infection followed by infection, that you make more robust antibody responses compared to infection or vaccination only. And going back to my initial slides when I was talking about rational vaccine strategies, we saw some of the germline V and D genes are highly enriched in these RBD antibodies, and also the S2 antibodies. So those could be used as templates to design vaccines that could work more effectively, and also B-cell immunofocusing to kind of avoid these antibodies that are frequently elicited but target non-conserved epitopes would be kind of an important area. So finally, my conclusions are that mRNA vaccines have been the most, if I have to point out one most remarkable progress in vaccinology in recent years, it would be mRNA vaccine platform. It has helped us to deploy these vaccines effectively and rapidly. Obviously, there are some questions still around how durable antibody responses do you generate with mRNA vaccines, and obviously we'll know more because these are new. We've conventionally relied on protein vaccinations or whole kill pathogens. So this will become more clear, but to rapidly respond to an emerging pathogen, mRNA vaccine platform has been remarkable there. So SARS CoV-2 is relatively, as compared to HIV, its spike is an easy target for neutralizing antibodies. You can get more neutralizing antibody responses. In HIV, in infection, natural infection, you get maybe 1 to 3% people that make broadly neutralizing antibodies. But in this case, it seems like more people may make them, and it makes sense that it doesn't have as effective glycan shield as HIV. As I said, through our studies and other studies from the field, RBD and S2 seem to be really promising targets for pan-sarbecovirus or pan-beta coronavirus vaccine strategies. And I think most likely we will need germline targeting strategies. Those are the rational vaccine design strategies, because we see a lot of enrichment of these genes in these broadly neutralizing antibodies that target RBD and S2. Immunofocusing strategies will likely be important, and I think probably like also NATO-like S proteins, those might be important. I think some of the major challenges ahead are, how do we generate mucosal immunity that can be long-lasting, that can protect us at the mucosal surfaces? I think the current vaccines are really effective at generating immunity in the blood, and it can travel to mucosae where you actually contract these respiratory viruses. It's not just SARS, it's a general problem for respiratory viruses, and we don't understand much about how to generate protection at mucosal surfaces. You need, some kind of IgA sometimes, and we haven't learned how to class switch our B cells with vaccines to generate those types of response. And I think going forward, I think that would be a very important area to consider, and to generate a long-lasting response, not just to coronaviruses, but respiratory viruses in general. So tracking variants of concern, I think it's extremely important area, especially in low middle income countries where the surveillance mechanisms for example, the genomic surveillance mechanisms are not that robust. So it's extremely important because sometimes, like in an immunocompromised individual, you could generate, these variants so effectively, like within a very short period of time, and then they start spreading. And then distribution and production of vaccines, and also like how we respond to variants. I think it's going to be a major challenge going forward, like how to update vaccines and stuff. So I would like to finally thank people in my lab who've done done this work on RBD. Wanting Hay, who was a postdoc, and Rami Mashrafi, who was also a postdoc in my lab. They drove most of the work on RBD with substantial help from Katharina Duker and Fabio Anzanello. All these studies were done in collaboration with Dennis Burton's lab, and Sophie Song, Pan Zhou drove most of the S2 work with help from Sean Callahan and Peter Young. Thomas Rogers and Davy Smith recruited all the COVID-19 convalescent donors for antibody isolation, and also Tom's lab and John Tao's lab have helped with some of the protection studies that I showed with S2 antibodies. We have a lot of collaborations with structural biology people here, and really feel fortunate that we have world experts here. Ian Wilson's lab, who did all the crystallization work, Andrew Ward's lab, who did all the electron microscopy structural work, Ralph Barrick's lab for doing those protection studies, and many other collaborators that have helped with different aspects of these projects. And my lab is supported by NIH and Bill and Melinda Gates Foundation. We have a substantial help from Scripps Chart that's led by Dennis and other supporters. And thank you for your attention. I'll take questions.